It is my absolute pleasure today to introduce Sara Nowinski, an assistant professor at Andel Institute in the Department of Metabolism and Nutrition, where she studies mitochondrial metabolism. However, I first met Sara when she was a postdoc in Jared Rudder's lab at the University of Utah, and she was just down the hall from me, and I was always impressed with her creativity as a scientist, her mentorship and her teaching, and also just her kindness. So this creativity in science led her to make fundamental discoveries on fatty acid synthase. Now, typically when you think of mitochondria, you think of it as only fatty acid oxidation and the breakdown of molecules. And what Sara discovered is that mitochondrial fatty acid synthase is a regulatory node for mitochondrial oxidation. And she characterized a new class of inborn errors of metabolism. Her works led to the receipt of numerous awards, including the American Cancer Society, the United Mitochondrial Disease Foundation, and the Pharmaceutical Research and Manufacturing Associations, as well as an NIH Maximizing Investigators Research Award, or AMIRA. Now, I am so proud to not only call Sara a colleague, but also a friend. Please join me in welcoming her, and let's hear some fun science.
Great. Thank you very much, Judy, for that kind introduction. And I've had a really awesome day getting to talk with people and meet new friends in this awesome department with some really cool things going on. And I'm excited to tell you about what my lab's been working on over the past 4 years at VAI. So, as Judy said, my lab is really interested in mitochondrial function and what mechanisms within the cell regulate mitochondrial function, because mitochondrial dysfunction is increasingly implicated in a whole host of pathologies, from rare genetic disorders like the inborn errors that Judy mentioned, to a lot of other diseases that are associated with physiological aging. And we really focus in on these rare inborn errors because we think that better understanding molecular functions and pathways can lead to better understanding of what goes wrong in these other physiological settings and provide windows into ways to target mitochondrial function in disease. And so since my lab started in summer 2021, we've really focused on these two questions. How do cells drive oxidative mitochondrial metabolism or function? And how do these processes go awry in disease? And we've approached these questions through the lens of the mitochondrial fatty acid synthesis pathway as a really critical node in mitochondrial metabolism that controls many of these oxidative metabolic pathways. So, most lipids in our cells, which you're probably familiar with, given the work that happens here, either come from exogenous sources or start with the synthesis by the cytosolic fatty acid synthase, which makes palmitate that's then further modified to make triglycerides, phospholipids, and our whole cadre of cellular lipids. But the pathway I'm talking about is actually not related to cytosolic fatty acid synthase at all, other than it catalyzes the same chemistry within the mitochondrial matrix. Instead, it's orthologous to type 2 bacterial fatty acid synthesis, and it's catalyzed by a series of discrete enzymes that make a growing acyl chain on an acyl carrier protein, or ACP. And each cycle of the pathway is going to extend that carbon chain by 2 carbon units. And when that acyl chain gets to be 8 carbons in length, it branches off through the lipoic acid synthesis pathway to make the lipoylated proteins. Lipoylated proteins are most famous for their role in the TCA cycle, as pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase both use a lipoyl moiety as part of their catalytic mechanism. However, it's been known since the late 80s and early 90s that this pathway could also build longer acyl chains in the mitochondrial matrix, and their identity and ultimate cellular destination is still not well characterized. But what I found as a postdoc in the Rudder lab was that these longer acyl chains were actually really important, crucially important, to support the assembly of the mitochondrial electron transport chain, as well as iron sulfur cluster biogenesis. And so you have in this mitofast pathway a way to link TCA cycle function through the enzyme lipoylation, as well as ETC capacity through this novel assembly pathway, and that they're both coordinately linked back to the ultimate mitochondrial substrate, acetyl-CoA. Now, as Judy mentioned, there have been pathogenic variants in mitofast genes described. Most commonly, the enzyme mutated is the terminal reductase in the pathway MECR. And also, there are now described mutations in the initiating enzyme MCAT, which is going to hook a mitochondrial malonyl-CoA to the acyl carrier protein to initiate a new pathway of elongation. These variants cause a syndrome known as MEPAN syndrome. It's a neurodegenerative condition that's characterized by a lot of the common features of mitochondrial disease, including dystonias, atonias, optic nerve atrophy, and balance and gait issues, as well as in some of the more severe cases, developmental delays and cognitive dysfunction. So we study this mitochondrial fatty acid synthesis pathway in a variety of contexts. We're really interested in how the pathway is regulated because really nothing is known. This pathway is incredibly understudied. And so we don't well know on the transcriptional, post-translational, or metabolic level how the pathway is regulated in cells and in different tissue types, as well as the consequences of impairments in mitofast function in tissues outside the nervous system. And so we know it gives rise to neurodegenerative disease in patients, but we've also shown in my lab that there's primary effects on skeletal myogenesis in the skeletal muscle stem cell population. And I'll tell you a little bit today about this cell type in the middle, and that's effector T cells. So I want to tell you a little bit about our work in the regulatory realm and in really just enzymes and pathways that are required for mitochondrial fatty acid synthesis, and then we'll move to the T cells in the second half of the talk. So it turns out it's difficult to study regulation of the pathway because the initiating steps in mitochondrial fatty acid synthesis are still really poorly defined in mammals. So I have it drawn here on this mitochondria that acetyl-CoA just becomes malonyl-CoA. But actually the source of mitochondrial matrix malonyl-CoA is not well understood or described. There's one paper that says maybe there's a matrix localized form of acetyl-CoA carboxylase 1 or ACC1. There are other papers that say that actually maybe malonyl-CoA doesn't come from acetyl-CoA at all, it comes from mitochondrial malonate. And if you knock out either of those enzymes or ACC2, you have no effect on pathway endpoints like protein lipoylation at all. And so we decided early in my lab to start trying to look for enzymes that are involved in initiating the pathway through CRISPR screening. And so we tried a bunch of different conditions and ran a bunch of different CRISPR screens using Kavanjber-Soy's metabolically targeted CRISPR guide library. And that all of this work was done in C2, C12 skeletal myoblasts under proliferative conditions. And it doesn't really matter what the two cell lines I'm showing you here. This is beta scores, which gives you an idea of guide enrichment or depletion along with significance of the observation. And what you can see is that under conditions where mitofast is required for cell growth, one of the top things that often falls out is NDUFA-B1, which is the gene name for the acyl carrier protein itself. And somewhere nearby NDUFA-B1, regardless what cell lines we were looking in, was always this other gene, AASCH-PPT, right? Here's another screen. Here you see NDUFA-B1, that acyl carrier protein, and here pretty close by you see AASCH-PPT. AASCH-PPT is a gene that encodes an enzyme called aminoadipate semialdehyde dehydrogenase phosphopantetheinyltransferase. If you can say that 5 times fast, I'll buy you a beer later. So, you know, a lot of people maybe would have looked at these data and that gene might not have jumped out at them as something super interesting or exciting. But when my lab saw this data, we were like, actually, maybe there's a project there. Maybe there's something to follow. And that's because of this. So like the acyl carrier protein domain on fatty acid synthase in the cytoplasm, the mitochondrial acyl carrier protein has a 4' phosphopanthenyl group. This cofactor gets added to a conserved serine on the acyl carrier protein, and it's the terminal sulfhydryl of that 4' phosphopanthene where the acyl chain actually gets built on the acyl carrier protein. There are actually 6 proteins in mammalian cells that get modified in this way. As you can see, there's FASN in the cytoplasm, AASDH, our protein's namesake, and then ALDH1L1, as well as L2 in the mitochondria and DARS2. So we know that these mitochondrial proteins have a 4' phosphopanthene modification, but how they get that modification actually wasn't known. So yeast have 2 encoded PPTase genes, PPT1 in the cytoplasm and PPT2 in the mitochondria. And it's also well known in yeast that these 2 cannot compensate for one another. So loss of PPT2 is incompatible with respiratory growth, and you get total shutdown of the mitochondrial fatty acid synthesis pathway. PPT1 cannot compensate at all. In mammals, AASDH-PPT is actually the only annotated PPTase in the genome, and it was unclear what was responsible for modification of the mitochondrial phosphopanthenylated proteins. And so we wondered, what is the mammalian mitochondrial PPTase, and could it actually be AASDH-PPT, even though we know that PPT1 can't compensate for loss of PPT2 in yeast. So to ask this question, and the alternative hypothesis is that maybe there's an as yet undiscovered mitochondrial PPTase. So to ask this question, we made CRISPR knockout clones for AASDH-PPT. And you can see these lines denoted here by PPT1 and PPT2. These are 2 separate clonal cell lines. I said knockout, but it turns out C2, C12s are near tetraploid. And actually in our PPT2, we only have 3 copies of the gene knocked out here. So these PPT2 cells maintain a little bit of residual expression, and you can see that on the blot there. And then we asked, does this have any consequences for mitochondrial function? So one of the easiest ways of doing that, as I'm sure you're all familiar, is a seahorse mitochondrial stress test, and that's what you see here. When we put the PPTase cells in the seahorse chamber, they have very little basal respiration and severely blunted maximal respiratory capacity. We then wondered if this was because of a loss in mitofast function or by some other indirect means. And so to do that, we can really easily look at this lipoic acid readout of the mitofatty acid synthesis pathway using Western blotting. So what you'll see here is we can actually blot for the lipoic acid modification on DLAT and DLST, which are the lipoylated subunits of pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase, respectively. And we see a pretty sharp blunting of that in our PPTase deficient clones, as well as a rescue when we add back a GFP tagged version of the enzyme. What we also noticed was that we had a decrease in the levels of SCHB stability, which I don't have the data in this presentation today, but we went on to show is a complex assembly phenotype similar to what we see in mitofast mutants. And then we also saw this shift in how the acyl carrier protein, or NDUF-AB1, itself was running on the blot. And we've seen in previous experiments targeting PPT2 in yeast that we would see a similar shift. And so we wondered whether these 2 bands corresponded to the holo or phosphopanthenylated form of the acyl carrier protein or the apo form of the protein. Oops, it's going to pop up with some boxes there. To go about asking that question, one of the first things we did was mutate that conserved serine to an alanine, and you can see that we observe a similar shift on the blot when we prevent phosphopanthene attachment by mutating that serine. And we also don't get really a further shift when we do that and express that construct in the PPT1 mutants, implying that we don't have a further loss in molecular weight. But we wanted to get at whether that shift was really phosphopanthethinylation in a really direct and targeted way. And so we came up with an IP mass spec strategy to assess the modification on the acyl carrier protein. So to do this, we expressed a flag tagged acyl carrier protein in cells and then pulled down that flag tag and digested with an ASP-N endoproteinase that's going to cleave on either side of that conserved serine at 2 conserved aspartate residues, leaving a DSL peptide, that 4-phosphopanthine, and then any acyl chain that might still be on. We can then shoot this on the IDX and we can monitor what comes off and look for what proportion of ACP is modified in each of our cell lines. So again, if we have an unmodified ACP, we're going to have just that DSL peptide. If we have a holo ACP, we're going to have DSL plus that 4-phosphopanthine, and then any number of acyl intermediates are going to come off. But this is showing you for the octanoyl or 8-carbon chain intermediate, we can see that species as well. When you run this on the mass spec, this is what this looks like. And the peaks that we get off map really nicely to the expected molecular M/Zs for these species. And so we can see apo, holo, as well as the octanoyl ACP. And we can also see them really well depleted in the PPT deficient cells, as well as a buildup in the unmodified acyl carrier protein. So this was really good evidence that indeed AASDH PPT was required in cells for the phosphopantetheinylation of the mitochondrial acyl carrier protein. So our question therefore became, where is this modification taking place? So what was known in the literature, as I've mentioned already, is that in yeast plants, cytoplasmic PPTase cannot compensate for loss of mito-PPTase. However, there was this paper in 2003 saying that greater than 90% of all the cellular activity of this enzyme was recovered from the cytoplasm when they were doing just biochemical fractionation and in vitro enzymatic type activities. And so we really weren't sure. We were pretty skeptical that it was happening pre-import, but you know, mammals are not plants and yeast. And so maybe they have a way to do this. So the first thing we did was just start out with simple immunofluorescence experiment where we targeted PPT or just expressed a mitochondrial DS red marker. And what I'll hope you can appreciate is just even with this endogenous antibody, we had pretty strong co-localization with at least a portion of mitochondria. Can you guys see that or not really? Is it possible to turn off the lights? No? Maybe. Okay, the next ones will be a little bit more hit you in the face. So when we express the PPT-GFP construct, it really strongly localizes to mitochondria and overlays with other mitochondrial markers like ATP5. And so hopefully you can appreciate that just by microscopy, we're seeing really good co-localization of PPT with mitochondrial markers, much different than what had previously been seen. Now, we can't tell from microscopy whether this is on the inside or outside of the mitochondria, though. And so we did a proteinase K protection assay, and this is an assay where you expose isolated mitochondria to increasing concentrations of digitonin to sequentially permeabilize first the outer mitochondrial membrane and then the inner mitochondrial membrane, and matrix proteins are the most resilient. And you do this in the context of a proteinase, which is going to be able to degrade proteins as they get exposed. And so what we saw was that if you look at an outer membrane protein, it gets degraded pretty immediately by the protease K. IMS proteins are more exposed, and then matrix proteins are the least vulnerable. And when we looked at that PPT flag signal, we could really see that it's maintained up to really the highest concentrations of digitonin, leading us to speculate or to conclude that this is truly inside the mitochondrial matrix. But you know, these biochemical assays are tricky, they're touchy. We had to do this and get the conditions to be just right. And so we weren't very confident in it. So it's always great, and this is a great thing for students to learn, that if you can show something in more than one way, then that becomes a much, much stronger confident conclusion. And so to do this, we took advantage of a system that's called bimolecular fluorescence complementation, or BIFC. And so we can express each half of a split venous tag on two different proteins of interest and ask whether they're in the same cellular compartment. And so what we did is we put the C-terminus of the venous tag onto the mitochondrial proteins with known localization, so mitofusin 2 on the outer mitochondrial membrane, or citrate synthase in the acyl carrier protein itself. As a control, we know PPT should likely interact with inside the mitochondria. And we also put this tag, the N-terminal, onto PPT on either its N-terminus or C-terminus. When we do this and then co-express all of the pairs in combination, what you'll see is that first, if we express C-terminally tagged PPT with that N-terminal venous, it does in fact only fluoresce significantly with citrate synthase in the matrix, or actually less well even with the acyl carrier protein itself. And so this to us, in conjunction with our proteinase K assay data, was really good indication that we were in fact seeing PPT actually inside the mitochondrial matrix. Now, what you'll notice is the same thing didn't happen if we put that tag on the N-terminus of PPT. And so instead, if we put that tag on the N-terminus of PPT, we now saw interaction with mitofusin 2 on the outside of the mitochondria. And so that also was really intriguing to us, because if you've studied mitochondrial proteins in biology at all, you'll know that the vast majority of them have N-terminal mitochondrial targeting sequences that will target them for the TIM-TOM import machinery and help the protein get into the mitochondria and then get cleaved as the protein enters. And so we wondered if this accumulation of the N-terminally targeted PPT on the outside of the mitochondrial membrane was indicative of an N-terminal targeting sequence on AASCH PPT. We ran the sequence of PPT through a number of different prediction algorithms that exist. One's called MitoFates. And it would come back with a weak prediction, like 40% likely to be a mitochondrial targeting sequence. That's not nothing. When we run totally non-mitochondrial proteins through the same prediction algorithms, they would be 0. But it's not the strongest either. And so we decided to just give it a try. And we made N-terminal truncations just to the next 2 methionines that occur in the endogenous sequence. And so this was at positions 13 and 36. And we asked, do these 2 truncation mutants have the ability to mitochondrially localize still? Okay, so here's our full-length PPT GFP again, and you can see they really cleanly overlay in the mitochondria with ATP5A. And then when we mutate just the first 13 amino acids, we suddenly have cytoplasmic accumulation and even a lot of nuclear-free GFP getting cleaved off the PPTase. In contrast, that 1 to 36 truncation just really isn't super stable. It's really difficult to get it expressed well, and so it's really hard to make conclusions about localization there. We then asked the finally really important question of, is that mitochondrial localization required for phosphopantetheinylation of ACP? Is it required for mitofast activity? And so to do that, we took advantage of our PPTase deficient cells, and we re-expressed these constructs and looked for rescue of protein lipoylation, right? So if we put full length back into the PPTase deficient cells, we can see recovery of that lipoic acid signal. And then we're going to look for whether we're able to recover when we put in the truncation mutants that can't localize. And lo and behold, we really can't. So we took this as good evidence that you actually need that mitochondrially localized PPT to be able to get ACP phosphopantetheinylation and mitofast pathway function. Now, I mentioned before that we're really interested in inborn errors in metabolism, and there are other mutations that give rise to disease in mitofast pathway enzymes. And so we wondered whether there could be pathogenic variants in AASDH-PPT that also give rise to similar pathologies in patients. And so we looked in the ClinVar database, and we came up with a list of point variants that were predicted to be pathogenic or predicted to cause genetic disease, but hadn't been characterized. And we decided to focus on the first 6, starting from the N-terminus of the protein. There was one that we were very excited about in that predicted MTS, and then 5 more in the CoA binding domain. We took advantage of the same rescue-based assay in our PPTase deficient cells and expressed these point variants and asked whether they had similar ability to rescue mitofast function and mitochondrial respiration in these cells. What we found over a host of experiments was that many of them had reduced function, so reduced ability to rescue protein lipomylation, and maybe perhaps even more clearly, reduced ability to rescue mitochondrial respiration when expressed compared to the full-length wild type. And so key takeaways of this first part are that AASDH-PPT is required for the mitochondrial fatty acid synthesis pathway, and therefore protein lipomylation and electron transport chain function. There's a pool of this enzyme that localizes to the mitochondrial matrix via an N-terminal mitotargeting sequence, and that mitochondrial localization is required for activity in the pathway. And then novel pathogenic variants from ClinVar can show reduced pathway activity, and this could be a new group of disease-causing variants. We're really excited about this IPMS strategy because it also gives us the capability going forward to do things that we've never been able to do before. So remember, I started off by talking about that question of where the substrate for the pathway even comes from. Is it acetyl-CoA to malonyl-CoA? Is it malonate to malonyl-CoA? We can now perform tracing studies into that octanoyl intermediate, into that acyl chain, and ask where is the carbon coming from, and then combine that with genetic studies to look at which pathways are required and is it actually ACC1 and ACSF3, are they compensating for one another? Okay, we're doing pretty good on time, so I'm going to shift gears at this point and tell you guys a little bit about what we've been doing to look at the role of the pathway in non-traditional tissue types in T cells. But I'm wondering if we want to stop here actually before we go forward, and if there are any questions about the PPTase part, we can take a couple of them now. Or not, we can just keep going and take it. Yeah.
Wait, hold on.
So when you show the mutations and you have one in the first 13 amino acids, I was expecting like a mislocalization, but it seems to be one of the variants that has the highest activity too. So it's like, can you talk about that?
Yeah, we were really hopeful for that one as well, that it would be a mislocalized variant. When we look at it, even though it's a P to S mutation, it looks like that it's still very mitochondrial. And so we don't know what's going on with that variant, but it didn't turn out to have the mislocalization that we hoped it would have. The other variants that do have bigger effects are all in the CoA binding domain. And I don't think that that dysfunction is likely unique to mitofast at all. So we didn't go look at their ability to phosphopantetheinylate FASN, but I would expect it to be affected too. Yeah.
I had a question about the localization of the PPT enzyme itself between the cytosol and mitochondria. How dynamic do you think is the protein moving in and out? Or do you think there is a specific pool of the PPT, maybe it's a splice variant that is localized to the mitochondria versus the majority of it stays in the cytosol?
Yeah, great question. I don't think that there's a splice variant that is at least abundantly expressed in the cells. Whenever we were blotting, even for the endogenous form, it's not like we were seeing a doublet in whole cells that went away. We do see the shift when we fractionate cytoplasm to mitochondria, but I don't think that the mitochondrial form, when you're looking at the endogenous, is nearly as abundant as the cytoplasmic form. I think it's just a weak mitotargeting sequence. And so Natalie Naimi, who I know a bunch of you are familiar with, has some really great work right now looking at like what makes a mitotargeting sequence and what makes one strong versus weak. I think this is just an example of one that it's not a great mitotargeting sequence. And so you do get important mitochondrial localization, but just most of the enzymes still in the cytoplasm. Yeah. There's one more back.
No, Carlos, we're doing this.
If I understood correctly, you did proteomics on phosphopantetheinylation proteins, right? Earlier, you said that there were 6 known proteins that get this modification. Did you identify any new proteins that get this modification in your proteomics?
Our method is targeted for the acyl carrier protein specifically because we do an IP for that flag tag at first. We tried running just a proteomics experiment. The peptide is there, but it's not abundant enough to quantify all the different modified species of it. And so maybe as mass spec sensitivity gets better. The ID'd proteins were actually using radio-labeled pantothenic acid and then tracing it into protein modifications and cutting it out and doing some of the really wonderful old school biochemistry with radio labeling.
Thank you.
Cool. All right, let's talk about T cells. So T cells are an integral part of the immune system. And if you think of the immune system like an army that's going to attack pathogens and invaders and keep us safe, CD4 T cells are like the generals. These are helper and regulatory T cells that are going to direct the immune response. And CD8 T cells are like the soldiers on the ground. These are cytotoxic T cells that are going to kill infected cells or cancer cells. And what's known among both of these cell types is that they undergo a huge metabolic shift when they encounter pathogens. And so naive T cells that have never seen and are not responding to some kind of foreign antigen are mostly metabolically quiescent. They have limited nutrient uptake, and they have an OXPHOS dominant phenotype. However, as they see and undergo expansion in response to a pathogen or other antigenic stimulus, they're going to undergo a huge metabolic shift towards when they become an effector T cell. They're very, very metabolically active. They're highly glycolytic, and they still have increased glutaminylysis and increased mitochondrial biogenesis. It's not like they shut off OXPHOS. That is still incredibly important, but they're going to ramp up glycolysis in a massive way at the same time. And so when I started my lab, we moved in next to 2 immunometabolism labs at the Institute. And I had a flox-flox model for MCAT, the first gene in the mitofast pathway. And I started talking to those labs pretty early on about looking at this pathway in T cells because they have to undergo this paradigm. And what happens if you take away their ability to do this in a coordinated fashion, right? This is different than breaking an ETC complex. You're going to have all sorts of complexes as you broke complex problems that result from breaking one ETC complex in the chain and having, reactive oxygen species and leaking electrons. This is a coordinated way to kind of shut off that oxidative metabolic program in many ways at once, because like we've been talking about, mitofast provides you an input into both TCA cycle and ETC at the same time. And so I said, what would happen? Let's just first look at what happens to pathway expression in normal T cells upon activation. And so immunology has a lot of these really nice tools and really nice databases available. And so we looked first in a mouse, this is the human RNA-seq, which is from the DICE database. And you can see that between naive and activated CD4s, as well as naive and activated CD8s, you get huge upregulation of transcripts encoding several of the genes in the pathway. So MCAT, MECR, OXSM, the acyl carrier protein itself, all massively upregulated upon T cell activation. And this was true when we looked in proteomics data sets as well. This is a mouse proteomics data set showing you if this time to the right is increased in activated cells relative to the baseline, which 0 is naive CD8 T cells. It's true in both CD8s and CD4s. And the other thing that was interesting to us is that it seems to be a MYC-dependent phenomenon, which, with the exception of the A cell carrier protein, most of these upregulations were dependent on MYC being expressed in the mice. And so we therefore wondered, okay, this is the central pathway for oxidative metabolism. It's massively upregulated upon T cell activation. What happens if we prevent that? And what does that mean for T cell effector function? So, as I said, we have a FOX-FOX model for MCAT, which is this first enzyme that's going to hook up malonyl-CoA to the A cell carrier protein. And we cross that with a CD4 Cree mouse model. So, T cells, as they mature and develop, undergo this maturation stage where they're actually double positive. So by driving CD4, the Cree off the CD4 locus, we're actually going to knock it out in all T cells, not just CD4, but also the CD8 counterparts. And so we did that, and then we asked what happens to T cell function in both those CD4 and CD8 T cells. When we look just by proteomics, we don't unfortunately have a great antibody for MCAT. It only works in isolated mitochondria, and T cells are tiny cells that don't have a lot of mitochondria. However, we did proteomics, and we can see a pretty severe loss of MCAT and no detection of MCAT in the activated Th1s, which are a subgroup of CD4s. We can confirm this by Western blotting for downstream indicators of pathway activity. And so, like before, you can see loss of that lipoylated protein in both the CD4s and CD8s, as well as a decrease in SDHB as a surrogate for complex 2 assembly. When we do seahorse in either the CD4s or CD8s, this is showing you CD8s, we have a really severe blunting of both basal and maximal respiration. And we have a little bit of dropping in mitomembrane potential per mitomass in the CD8s that was more pronounced in the CD4 Th1 subtype. So this was pretty good evidence to us that these are all things we knew, things we had seen in the C2, C12s and the skeletal muscle cells and other systems. And so this is a pretty good indication to us that MitoFast is likely acting in a similar way in these T cells, doing similar things that we expect it to do biochemically. So now, what does this mean for T cell function? To assess that, we did a series of in vivo infection type experiments. And so this is showing you Listeria, which is a gut infection that is going to involve both a CD4 and CD8 response. And what you can see is that we can administer alum ova, which is a Listeria substrain, and isolate the splenocytes, isolate the T cells from the spleen about 7 days after infection, and then do immune phenotyping. And what you'll notice right away is that the MCAT knockouts have a reduced cell number at this time point, as well as a reduced number in activated cells marked by the percent CD44 positive high cells. And so I saw these data and I was super excited about them. Like, look, they can't respond and they can't activate in response to Listeria infection. And I met with my collaborators in the immunometabolism labs, and they were like, yeah, it's actually not a big phenotype for immunology. I mean, it's a phenotype, it's real, but I don't know if this is super exciting yet. And I said, okay, well, what do you think? How do we know, if this matters? And they said, what you really need to do is go actually look at T cell effector function, not just markers and numbers of cells. And so we can monitor T cell function via cytokine production. So the actual job of these T cells, both CD4s and CD8s, when they get activated, is going to be to secrete cytokines like interferon gamma and TNF alpha, and those are going to activate and recruit more immune cells and T cells that are actually then going to lead to cell killing and pathogen clearance via things like granzyme B and perforin. And so what we can do is we can take the cells out of the spleens that have seen the Listeria infection, and we can re-stim them with a peptide in culture that's specific to that Listeria antigen that the Elm ova strain exposed them to. And so we use a slightly different peptide, whether they're CD4s or CD8s, but we can re-stim with over LLO in culture. And so the cells will recognize that and start secreting more cytokines. And this will tell you if they're actually functional or not. When we do this, and then we use flow as a readout for interferon gamma and TNF alpha production, what you can see is that the MCAT knockouts are really unable to respond in this MCAT, in this peptide re-stim assay, either the CD8s or the TH1s. And so this is now a really severe functional deficit and likely to be impactful for their ability to clear the infection. Okay, so we show you this for Listeria. We can also show you this in response to viral infection, which is much more heavily driven, the viral clearance by CD8s. And so to do that, we use a mouse virus model called LCMV. And LCMV, similarly, we're going to infect with it. 8 days later, we'll isolate the T cells, and then we can do immune phenotyping or a GP33 peptide re-stim and quantify the cytokines. And similar to what we were seeing in Listeria, when we do that peptide re-stim in the MCAT knockout cells, we have reduced antigen-specific expansion, reduced cytokine production, and really severe blunting of function. And so we wanted to know why. What functions downstream of MitoFast are important for this phenotype? And so we did a lot of tracing data, a lot of metabolic analysis that I'm not going to show you today, that basically showed that even though we have blunted protein lipoylation in these cells, we don't have big differences in TCA cycle activity. So we don't actually think that this is due to loss of protein lipoylation, because actually my collaborators have shown that a lot of T cell derived TCA cycle activity isn't coming from glucose at all. It's not coming through that PDH derived pathway. T cells actually do a lot of ketone body metabolism. And so they love beta hydroxybutyrate as a substrate. And we think that that might be why we don't see huge TCA cycle deficits. Instead, what we did see was indicators of two other downstream pathways. So I haven't told you in detail about this ETC assembly mechanism, but it turns out that it's actually very well characterized at this point, and it involves two things. It involves physical interactions between the acyl carrier protein and a family of ETC assembly factors, as well as defects in iron sulfur cluster biosynthesis, which I'm going to call ISCs for this talk. So the iron sulfur cluster biogenesis machinery has a core subunit, catalytic subunit called NFS1, and then two other subunits called LRM4 and ISCU. And ACP binds to that LRM4 to stabilize it and this whole complex. We actually have structures of what this looks like, thanks to Dave Berendau's lab at Texas A&M. And so you can see in blue the acyl carrier protein. This colored part is actually that 4' phosphopantethine cofactor, and then an acyl chain that folds down into the middle of LRM4. And I think it doesn't take a ton of imagination to see why that acyl chain might be important for stability of that 3-helix bundle that it's wrapped into the middle of. And so we know that ACP plays an important role in iron sulfur clusters through this mechanism, as well as ETC assembly. And so when we kind of weren't getting anywhere with TCA cycle function, we wondered whether it was these longer acyl chains and these other parts of the pathway that were important. To start getting at this, we just did unbiased quantitative proteomics analysis, label-free in both our CD8 and CD4 T cells. And when we do go gene set enrichment analysis for Go terms or canonical pathways on that, we get back a lot of things that look very ETC-like, right? We further analyze these data by doing leading edge analysis on them. And you can see we start to get groups here that group by ETC subunits, ISC containing complex 1 subunits specifically, redox reactions, and other ISC interacting proteins, with this group in the middle for glycolysis and nucleotide metabolism. This was in the CD8s. When we look at the CD4s, the picture is even more clear. It's really, really clearly complex 1 and iron sulfur cluster biogenesis. And so we wondered, is this actually the main target of the mitofast pathway in the T cells that's giving rise to this phenotype? What does the ISC biogenesis machinery look like in these cells? To do that, we just simply blotted for the members of the complexes, and you can see we have a really stark loss of NFS1, LRM4, and ISCU in our activated CD8s and CD4s. If we further analyze our proteomics to look for iron sulfur containing proteins and complex 1 subunits, we get a really clear group of proteins that drops out in the TH1s. These are iron sulfur containing subunits of complex 1, which are also all over here on the left in the CD8s as well. And so this to us was really pretty intriguing, that this could be potentially responsible for our loss of CD8 and CD4 effector function. It's really difficult to rescue because you can't re-express LRM4 in the T cells without rescuing that acylation. But we tried to do it. I've tried to do it in the C2C12s. It really doesn't work. And so we took an orthogonal approach and said, what if we just CRISPR out NFS1, LRM4, ISCU? Is it sufficient to do the same thing as MCAT in naive T cells? This is showing you we can target either NFS1, LRM4, or actually ISCU is lost in all 3 of these cases. And we have a very similar phenotype to that we see in the mitofast knockouts, where they have blunted ability to produce interferon gamma. 2 different ways of looking here, interferon gamma high expressing cells or the MFI. Now, this was interesting to us because all of these data strongly imply that the T cells would not be able to clear infection in vivo. And that's difficult to measure. However, one thing that we also know is that T cells become exhausted in settings of chronic stimulation. And so when you have something like cancer or a chronic infection, the T cells really lose their ability to do their job. They become refractory and can no longer produce interferon gamma and TNF alpha. And I've already shown you we have that phenotype. And so we wondered, if we looked at markers of exhaustion, are we seeing enhanced T cell exhaustion in the MCAT knockout mice? To do that, we did both an in vitro chronic stimulation assay, and you can see, I think, pretty clearly that we have increased markers of exhaustion only in the chronic infection in the MCAT knockout mice. Controls have a bump over acute infection, but nowhere near the extent the MCAT knockouts do. And then we also can see this in vivo with a model of chronic infection. This is a different clone of that same LCMV virus that the animals can't clear, and so they're chronically infected with it. And in addition to that increase in exhaustion markers, we have really flatlined CD8 cytokine production in this chronic infection model. And so this second story that I've shown you is as yet unpublished, but it's under review right now. And the key takeaways of it are that mitofast coordinates iron sulfur cluster biogenesis and ATC function to support T cell effector function. That knockout of the pathway impairs T cell responses to a wide variety of infections. I didn't show it, but we also did Citrobacter, and we see very similar results. MCAT knockout impairs T cell. Yeah, I said that already. Cool. This is mainly via reduced cytokine production and driven by loss of iron sulfur cluster biogenesis. And that loss of the pathway promotes T cell exhaustion. So I'll end there. I need to thank my lab. So the phosphopantetheinylation story was really driven by a research scientist in the lab, Peter Norden, and Riley Wadan, who's a MD PhD student. And the T-cell collaboration has been really driven by Sam Preston, who came in as a postdoc, had this idea, really wanted to pursue it, and made it happen. So thank you very much again, and I'm excited to take more questions.
Questions for Sara on the second half of the story?
A really good talk. I might have missed when you drew this link, so just to ask for my own clarification, do you have any idea why the disruption of the empty fast pathway that you were just discussing would actually lead to the exhaustion? Is there like a link there that in disrupting that, what directly then makes that phenotype happen?
Yeah. So you can do these kind of complicated experiments where you can do adoptive transfer experiments in mice where you clear the immune system and then give them back a mix of wild type T cells and your knockout T cells and let them compete kind of and see. So when we do that, we see one, the wild type T cells way expand and outcompete the MCAT knockout. And they also, the exhaustion phenotype goes away. And so we don't think that it's an cell intrinsic effect where it's exhausting them like just on their own. We think that it is that they can't clear the virus or the bacteria infection as efficiently, and then that's leading to additional exhaustion. But we haven't looked in cancer cell settings yet, so Sam's really, really keen to do that for a second story. Yeah.
Thank you.
Yeah, I'm gonna jump in. So obviously you're focusing on T cells because of the kind of switch between a mitochondrial oxidation and glycolysis as they become more mature. I guess I'm curious though, are there other cell types that you think that this will be particularly important for? Because it seems like that switch is a critical point in the development of a lot of cells.
Yeah. So I didn't talk to you guys about it today because I already jammed 2 stories in and couldn't, but we see a really, really similar thing in skeletal muscle stem cells. And so skeletal muscle stem cells also undergo a really similar metabolic shift, right? They are proliferative and glycolytic as they repair tissue, and then they switch to predominantly oxphos heavy as they differentiate. And we, in our skeletal muscle cells, see massive problems with differentiation. And then we have a mouse model where we've knocked it out specifically in that skeletal muscle stem cell population, and they have reduced or impaired ability to repair muscle after injury. And so, yeah.
I guess I'm curious too then, because we know that that switch in glycolysis is important for like cardiomyopathies and heart failures and stuff. And so they go back to that like fetal programming and use of like reliance on glycolysis. Have you looked in, because you started off with this concept of disease states, have you looked at like this pathway as it changes?
I haven't looked in heart failure specifically. We've looked in some of the diabetes databases or models that are out there, and I think that there are differences in adipose tissue expression levels of the pathways in type 2 diabetes and things like that. But I'm really keen, even if the pathway is not dysregulated in those disease states, to know like this is such a central regulator of mitochondrial metabolism. Could we drive this pathway and use that as a means to sort of push mitochondrial oxidative metabolism in these disease states? So we did a bunch of work to figure out what the rate limiting enzyme in the pathway is. It's one I didn't talk about today called OXSM. And we made a mouse model that overexpresses that, and it looks like it can drive function above baseline. It's a mild effect, but we haven't looked in disease states where function is decreased yet. So that's what we want to do. That's actually what I wrote my last grant about. So yeah, thanks.
All right.
So in your MCAT knockout cells, do you have a sense for if the exhaustion is happening on like an accelerated timeline or if it's more just widespread across the T cells?
Yeah. So I didn't show these data today. It happens if we look, I showed the proper way of doing it, which is the chronic stem and the in vivo chronic infection models. You do see it come up faster in the chronic stem assay. This is probably the best way we've looked in a time-dependent manner. But I can also tell you we see it come up actually in the acute infection when it shouldn't be there at all, because normally the wild type mice, they can completely clear the acute LCMV infection. So the wild type mice there don't exhaust at all, and these guys do.
All right. If there's no more questions, let's thank Sara for her beautiful talk.